Each year, Microsoft Research hosts hundreds of influential speakers from around the world, including leading scientists, renowned experts in technology, book authors, and leading academics, and makes videos of these lectures freely available.
Okay, it's a pleasure to have Ernest with us. Ernest, of course, does a lot of great work in computational biology, and he's practically one of us, so it's really, really fun to have him speak.
Great, thank you very much. So I recognize some, but not everyone in the audience, and I recognize it's a heterogeneous crowd. So if things are not clear, you might as well interrupt me rather than sitting through the whole talk and nodding off the way I might do if I was in the audience. Instead, stop me quickly. So what I hope to do today is show you how the kinds of algorithmic work that's being done here at Microsoft and other places around the world, of course, can have a big impact on biological problems. And they have applicability to a lot of different biological problems, but I'm particularly interested in the biomedical aspect and in how this, coupled with other new technologies, could really impact human health. And give you a sense of ways in which technology can impact human health, I think it's a little historical perspective helps. This is a plot of mortality rates, the number of people who died per 100,000 of the population over the course of the 20th century. And there are a couple of trends that are really very striking here. The most striking one is the least relevant. It's the spike in 1918, which is, some of you may know, the Spanish flu. But what's really dramatic is this biphasic decline. First, there's a very steep decline in death rates in the first half of the 20th century, and then a slower rate of decline in the second half. And this was driven by technological innovation. Not always the most sexy technological innovation. So if you separate this into infectious disease deaths in red and non-infectious in blue, the infectious disease deaths are what are really accounting for the decline. And it starts with sanitary engineering and food science research that really made it possible to live in cities, and that dramatically caused a drop in infectious disease deaths. And then about mid-century, or what most people think of, the pharmaceutical industry and its development of things like penicillin that really continued the decline. So at the beginning of the 20th century, about half of all the deaths in the United States were due to infectious disease, and at the end of the 20th century, regardless of the fact we had the AIDS epidemic, which is this slight little blip here, there's really almost no one who's dying of infectious disease as a fraction of the total. So technology can have a huge impact on human health. Now, we've done really well as a society in conquering infectious disease in the developed world. We've not done so well in dealing with non-infectious disease causes. The rates of death are pretty much the same over the course of the entire 20th century, despite the fact that billions of dollars are being poured into the pharmaceutical industry. And so the question is whether there's a new approach to biology, different from what was going on from over the course of the 20th century, a 21st century version of biology that can get us there. And this is where, yes?
This particular slide, what happens in developing countries? I mean, is the plot similar? Or?
I haven't, so these are not my data. This is from the JAMA journal, and I don't actually know what the plots look like for the developing countries. I don't think there was such a steep decline in infectious disease deaths. I think it's been much more stable, unfortunately. So,
another
yeah.
unrelated, but so AIDS basically unnoticeable on such scale of
AIDS, yeah. In the United States, right? If it was Africa, it would be very, very different. Yeah.
Where are you expecting the blue line to go? Given
Well, it can't go to zero, right? Can't go to zero, but you might expect people would both live longer, and so you could have, this is deaths per 100,000 of the population, so that number can drop. It's not going to go to zero, but it could drop. Okay, so for a lot of the 20th century, biology has been driven by somewhat anecdotal approaches to biology, where someone stumbles across a certain molecule, discovers that's important, and then tries to understand its larger significance. And that's driven a lot of pharmaceutical development as well. But what we're on the verge of now is a very different approach, which uses a lot of different high-throughput measurements to systematically look at different kinds of molecules inside the cell, and then computational models to help us understand what's going on in the cell in a way that could predict where we should intervene. And there are a lot of these measurement technologies. The oldest of them are technologies that measure RNAs in the cell. There are new technologies that measure proteins and the way that proteins interact with each other and the way they interact with the genome. And they're called omic data for short because of genomics, transcriptomics, proteomics, all these different omic technologies, just a shorthand for measuring as much as you can about what's going on in the cell, so that in principle, you could measure tens of thousands of data points about each cell in a disease state and in a healthy state, and then try to reconstruct, develop clear strategies for how to intervene. So in order to make this understandable, I'm going to do a little digression, sort of everything you need to know about molecular biology in about 2 minutes. I don't know if you're familiar with this for Dummies series, but I like to think of it as books for smart people with very little time. So I'm going to give you the molecular biology for Dummies course here. When we think about biology, we can think about it in many different scales. But fundamentally, a lot of biology deals with how cells and organisms respond to external stimuli. So you could think of, for example, of toxic compounds or mutations or infectious agents. And those obviously can have very macroscopic effects on biology. So animals can move, cells can move, both animals and cells can die or grow. But we're very interested in the molecular changes that underlie that. So the kinds of molecular changes we're interested in, every gene in the genome doesn't get turned into RNA and then ultimately into protein through the processes of transcription and translation. What happens to those proteins? Do they get modified? Do they get moved around in the cell? Do they get broken down? And one of the kinds of modifications that I'll talk about later, so I'll introduce this here, about that happens to proteins is something called phosphorylation. And it's a simple chemical reaction that changes an oxygen and hydrogen to this thing that has a phosphate and other oxygens. So this is called a phosphate group. There are proteins that carry out this chemical reaction, and they have, unfortunately, names that are not intuitive. So the protein that adds the phosphate group is called a kinase, and the one that removes it is called a phosphatase. This chemical transformation is particularly important because a lot of proteins, when they undergo this change, will go from a state where they're either active to inactive or inactive to active, or they can be designated for being moved around to different parts of the cell based on this phosphorylation reaction. So understanding which proteins are getting phosphorylated and what happens to them when they get phosphorylated will play a big role in what happens later. Here's a cartoon of some of the things that can happen. So if this is the barrier between the outside of the cell and the inside of the cell, and various signals are detected from the outside of the cell, and then that information gets transmitted into the cell. So there'll be proteins that span the membrane on the surface of the cell. They detect these molecules. And then one of the things that they can do is get phosphorylated, that reaction that I told you about in the previous slide. And that can cause two proteins to interact that would not have interacted previously without the phosphorylation. And it can also change reactions so that one of these proteins that gets phosphorylated can then cause other proteins to get phosphorylated in a chain reaction that then ultimately leads to something macroscopic that you can observe. So this is a kind of pathway that occurs when information goes from the outside of the cell to the inside of the cell. And those signals then often go through the cytoplasm into the nucleus of the cell, where they influence how genes get transcribed. So this will be the symbol I'll use for a gene. And under some conditions, the gene gets turned into RNA that ultimately gets made into protein. Under other conditions, it's not. What determines whether a gene is active over here or inactive over here is whether these regulatory proteins find the right gene in the genome, sit there on the DNA, and send the signal to transcribe them. And so the signal comes from outside the cell. It goes through the kinds of phosphorylation changes I showed you on the previous slide. Those eventually get to the nucleus, where they cause these proteins to change their binding or change their activity, and then you get genes transcribed. Okay, so that's the core signaling process. And here's an example of what one of these processes looks like. So you can see this looks pretty complicated, right? There are a lot of signals that are coming from outside of the cell. These have to do with insulin, which changes after you eat a meal. They go through a bunch of different proteins that ultimately go to these complicated processes that are so complicated they're not even represented here. They're just shown by words. So the cells take up more glucose, or they make more proteins, and so on. Now, the problem is that while this looks complicated, it's actually a gross simplification of what's going on in the cell. And in fact, a lot of what goes on in the cell we don't know about yet. And so what we'd like to do is stop reasoning by drawing pretty pictures and start reasoning on the computer. And that's where the algorithmic approaches really come in. Can we take a much more systematic approach, find all the genes and proteins involved in these complicated processes, and let the computers tell us what's important, rather than saying, oh, GSK3 looks really interesting, I'm going to spend a career proving that GSK3 is important for insulin resistance or cancer or whatever it is. So that's the motivation. We're going to measure as much as we can about what goes on in the cell and computationally figure out what's important rather than trying to do it all in our heads. So that's the goal, and what I want to do in the rest of this talk is explain some of the challenges that we faced when we try to take that comprehensive approach and really unbiased by our preconceived notions, and then show you two things that we need to be able to do to really integrate everything. One is focus on that process of transcriptional regulation, which genes get turned on and off, and then assembling larger network pictures of the whole signaling process. Okay, so let's begin with the challenges. So the first challenge that we face when we try to take a very systematic approach to these biological problems is we find that a lot of the responses are not at all what people expected. So people have been studying some of these signaling pathways for decades, and they've built up those nice pictures that I showed you before. But when you actually do a specific experiment to test what the response is in the cell when you perturb those pathways, you typically find that only about 10 to 15% of the response is in the expected pathway. And a lot of the response falls in sets of proteins and genes that are not annotated as being involved in any pathway. People just don't know what they do. They may know some specific aspects, so they don't know the larger context of what they do. So this presents both challenges and opportunities. The challenge is that if you get data from some very systematic measurement and you plug it into some program that says, what do people already know about it, you're going to tend to get the wrong answer, right? The programs will tell you, oh, well, this was a perturbation to one of these pathways, and in fact, it was a perturbation to this pathway that constitutes 10% of the signal. The opportunity, though, is that we obviously are, as a field, been taking too narrow a view of what's going on if we only call 10% or 15% of the response this pathway and call everything else something else, right? And there's a lot of this we don't know anything about. So it could be that if we had a better understanding of what the 85 to 90% of the response is, we could design better therapies. We'd understand the more larger scale context of what's going on. Okay. So this first challenge seems like mostly opportunity, not too bad. The second one seems a little bit more daunting. This is what we find when we try more than one experimental technique to analyze the same problem. So here's just a cartoon showing some cell that's been exposed to a perturbation from which we've measured different things. We've measured which genes are changing in expression and which proteins are changing in their amounts, and which genes, when you knock them out in the cell, make them inactive, have an effect on the response to this, say, mutation or environmental change. And the Venn diagram of the hits from each of these assays, the things that are significant, looks something like what you see on the right here. So there's rarely any overlap between different data sets that are measuring ostensibly the same thing. In fact, we've found that it's usually less than you'd expect by chance. Okay. So you're all laughing. You should be concerned, right? So why is it that you get less overlap than you expect by chance? Now, one possibility, the pessimistic view, is that these assays are just terrible, right? And they're just all noise. But we know that that's not the case because each of these assays independently is highly reproducible. So if I do this assay more than once, I'll get a Venn diagram that has 80, 90% overlap. So why is it that when I do this assay compared to this one, I get so little overlap? So that was a question that two postdocs in my lab at the time and now have their own labs in their home countries tried to solve, Estier Garlotum and Laura Riva. And they were looking specifically at two kinds of experiments. One is this gene expression, what genes get increased in expression or turned down when you expose a cell to some perturbation. And the other one, if you knock out the gene, make it inactive in the cell, what happens? Does it have a phenotype? Does it affect how the cell grows? So what they did was they looked at lots and lots of data where they had both kinds of experiments, the gene expression and the genetic data. And just like on the previous slide, there was almost no overlap between the different data sets when they were conducted in exactly the same setting. But what they found was there was a simple explanation, which was each of these kinds of experiments was biased towards a different type of molecule. So in this case, the genes that change in expression tend to be what they call effector molecules. They're carrying out some function. Whereas the ones that were detected in the genetic screen when you knock them out, those were master regulators. And I'll explain what I mean with a specific example. So if you look at cells that are exposed to a DNA damaging agent, and you look at which genes the cell turns on when it's exposed to DNA damaging agent versus which genes, when you knock them out, affect the ability of the cell to live in the presence of a DNA damaging agent. So we get two very different answers. if you knock out certain genes, like the ones that detect DNA damage, then the cells die when exposed to DNA damaging agent. So these detectors of DNA damage, they actually run across the DNA. They say, oh, there's been DNA damage here, and they start a response pathway. These show up in the genetic data, but they don't actually show up as differentially expressed. They don't get turned on by the cell. Now that actually makes sense, right? If you think about this analogous to, say, a smoke detector, you keep the smoke detector on in the house all the time. It doesn't go on after the fire occurs. It's on before, during, and after the fire. So the DNA damage detector has to be on all the time. It's not going to show up as changing in expression in response to DNA damage. By contrast, things that affect the response, things that make the cell stop going so it can repair the DNA damage, or the enzymes that actually do the repairs, those get turned on specifically when you need them. They're not on all the time, the same way that, say, these sprinklers are off now, but were there to be a fire, hopefully they would turn on. Now, why don't these effector molecules show up as genetic hits? Well, for the same reason that if I knock out one sprinkler in the room, we'll probably be okay if there's a fire, because there's redundancy. So there's a lot of redundancy, these effector pathways. So when we have discrepancy between two different kinds of data, it's not always because there's just noise in the processes. Sometimes they're asking very different questions about what goes on. If you understand the question, then you can understand what the answer is, right?
So if I understand the analogy right, there were two different kinds of molecules, and we were thinking of them as the same. And now these kinds of things, like experiment A was measuring A1, this is measuring A2.
Exactly. So the different kinds of experiments detect different things, and there are some molecules that are on the same pathway that aren't detected in either experiment, that people have done other kinds of experiments that detect. And so this gave us the idea that maybe a way to reconstruct these complete pathways would be looking for connections between the things we get from one kind of experiment and the things we get from the other kind of experiment. And along the way, then we pick up some of these other things. So in a cartoon sense, what we're going to do is we're going to have some cartoon of the cell here. And this is the outside of the cell and some detector molecule, and this is the nucleus. And I've painted things with different colors based on the different assays that pick them up, and nothing gets more than one color because, in general, we rarely find anything detected by more than one assay. But there could be some pathways. Maybe no one's called it a pathway before, and those we'd expect to have some molecules detected by one technique and some molecules detected by another. So we'd like to develop algorithms that allow us to reconstruct these previously unknown pathways. And then along the way, we'll get information about proteins that weren't detected in any of the assays.
So measuring the same process with different measuring techniques, but not simultaneously.
You can do it simultaneously. That doesn't really influence what you're detecting here. But that's not the explanation for what's going wrong. It's the bias of the types of experiments and the types of molecules.
Right.
So our thought is that we can change people analyzing data. Plug into databases that have lists of what's already known about molecules. But I showed you that that's misleading, right? If you just look for the effect pathway, you'll be misled. Another thing you can look for is a little bit of overlap between different assays, and that's also not going to get you the bulk of the data. So we'd like to take an approach that really tries to reconstruct the relationship among those two types of data. And so that's what the rest of the talk is going to be about. So the idea is we're going to map our data onto a network of known interactions known molecules. Yeah.
Go back a slide. So the overlap represents pathways in which both versions of these things play a role. Is that what
So
we should think
most
about?
pathways will have influence from all these kinds of molecules.
And the overlap, was it something that we could have sort of pointed to saying, oh, there's a blue and a yellow thing on the pathway, then that will sort of show up in the overlap? Or
Oh, so the overlap is the same. Would the same molecule be detected by more than one assay? And that tends out to be very rare. But we think that most pathways will have contributions from all the different kinds of molecules.
Okay.
So what do your data look like? Do they look like various experiments and assays, and then are these pathways like biophysical models or something like this? And you just have like these tables of disjoint data sets or something? Like I'm trying to figure out.
Yeah, so the kinds of data, this might become a little bit clearer as I go through, but the kinds of data you'll get would be for every, say, gene in the genome or every protein that's in the proteome, you'll get some number that represents the, say, the amount of change between two conditions. And then you can assign a statistical significance to that and say, here are a set of, out of the 20,000 genes in the human genome, these are the ones that seem to be statistically significantly changed between condition A and condition B. And so that might be for this, and then you'd get a similar list from which proteins seem to be changing at the protein level, which genes, when you knock them out, seem to be changing the viability of the organism. So you'd have long lists.
And these lists are put together in pathways because you have sequences.
Well, so the pathway reconstruction is really what we want to accomplish. So there are some pathways in the databases. Those are like those pictures I showed you at the outset.
Yeah,
Those are very incomplete.
So you have these tables of these kind of individual statistical interactions, then you have these databases of chains of events.
That's right. And then there's another kind of information that I'll bring in a little bit that represents the links between proteins that may not have been annotated as a pathway yet, but do represent physical links among these proteins or between proteins and genes. Yeah. Other questions? These are good questions. Okay, so we want to be able to find the connections among all these different types of data. The risk is, though, that we get these giant networks that really are uninformative. We lovingly in the lab call them hairballs, right? In a lot of these algorithms, you take a small data set of things that came out statistically significant from these assays. Maybe there are 20 genes and 30 proteins, and then you end up with a network that has 300 or 1,000 or 10,000 genes. And it's very hard to figure out what you would do with that that would actually lead you to deeper biological insight. So we need to use some algorithmic cleverness here. So our approach then is going to be to try to find these networks, but try to find compact ones, and that's where a lot of the work goes in. The other thing that's important to know about the approach has to do with how we treat certain kinds of data. So some of you may be familiar with the idea that biology has what they call the central dogma. I always say you should be afraid of any field where they claim they have dogmas and they're proud of it, right? But the central dogma of biology is that DNA gets converted to RNA, which gets converted to protein. And it turns out it's much, much easier to measure changes in RNA levels than it is to measure changes in protein levels for various experimental reasons. So for a long time now, people have been assuming that changes in RNA levels were decent proxies for changes in protein levels. So I measure something systematically about all the RNAs in a cell, and I say, okay, well, that must be true for the corresponding proteins, and therefore I can do something about the consequences for the cell. Recently, people have been actually able to experimentally test whether that's true, and it turns out that it's a really, really bad approximation. So on the x-axis are RNA levels, and on the y-axis is the corresponding protein level for each of those genes. And you can see that, yeah, sure, there's some crude correlation, but there's a huge range of variation, 3 orders of magnitude for all the RNAs at the same level.
I mean, if you did sort of, I mean, there's a variance issue here too, right? It could be like this is the right mean level. Are these sort of from different experiments, or is it sort of the same condition repeated?
Same condition, yeah. So it's not an issue just of noise. It's an issue that there are regulatory processes. So once you've got the RNA, it doesn't automatically get turned into a protein, and not every RNA gets turned into protein at the same rate. And once it does get turned into protein, some of those proteins get broken down quickly, and others last a long time. And that's what causes the 3 order of magnitude variation. So for the longest time, people had been saying, okay, RNAs get turned into proteins, so my RNA data, which is quite abundant, will serve as a proxy for whatever the consequences are, the increase in the proteins. And that turns out to be a really bad idea. So then we're faced with this problem. There's a lot of information about RNA levels. What are we going to do with that? Right? Are we just going to throw it out? So, yeah.
So is that each of those points were from a different protein, right?
Each data point is a different protein, right? Yeah, exactly. So what do you do with it? Well, it's terrible proxy for this, but what actually is a fairly direct readout is of this process that turns the genes on. So if I have more of a transcript, it means something turned that gene on, right? If the lights go on, something was touching the light switch. So what was touching the light switch? It was these proteins that bound to the genome and changed either their binding or their activity. And so we're going to try to use those RNA levels as evidence of this proximal upstream process rather than these downstream things that are badly correlated. And so what I'll talk to you about today is how to actually do that, to take the gene expression data, which will be represented down here by these boxes, and use it to map back to the DNA binding proteins that sat on the genome and said, turn on this gene or turn this gene off. And then once we've been able to do that, then we'll look for connections to other kinds of data. So we'll then have data from other kinds of experiments about proteins, and we'll look for the links up to that. So we'll take this bottom-up approach, gene expression, to the things that turned on the gene expression, the DNA binding proteins, the things that turned on the DNA binding proteins, all the way up. Okay. So in order to do that, we need to get good at two things. One is figuring out this transcriptional regulation question, which proteins are binding to which genes and when. And after we've done that, we can then try to reassemble this network model. And so what I'll show you is just a little bit of how we do each of these steps. Okay, so there's one way you can measure protein-DNA interactions, which is basically cheating. You can actually do an experiment, one protein at a time, figure out where it binds in the genome, and that is called genome-wide chromatin immunoprecipitation. It's kind of a long name. It's been done successfully. We did it with a guy named Rick Young at the Whitehead Institute for almost every protein that binds to the genome in yeast. It turns out that it's a lot easier to do this in single-cell organisms like yeast than it is in humans and in mice. It's virtually impossible to do this on a global scale. So even this giant multi-institution effort to map out all the things has only covered about 10% of all the transcription factors, these DNA binding proteins, and only under very specific conditions, probably not under the conditions that any scientist looking at a particular disease would be interested in. So people have tried various computational approaches to fill in the gap. And one idea is, if there's some change between an active and an inactive state of the protein, well, maybe it's, well, it must be because there was some change in the regulators, the light switch turning on or off. And one thing that could change the regulators would be if they themselves are changing their amount of transcription, right? So if the gene that encodes this dash protein is off, then the dash protein isn't made, it's not sitting in the genome, and then its targets will be off. So people have looked in the gene expression data itself for the evidence of the regulators and tried to build a self-consistent model of what's going on. And there's a lot of literature on this. These representations you'll see, usually the red boxes represent a gene on the rows and conditions on the columns. And if it's red, it means that the gene is increasing in expression in that condition. And if it's green, it's decreasing. And so people look for these self-consistent models where there's some protein and it's on under these conditions, and that's why these are on. And so on and so forth, activators, repressors, and there's a huge literature on this. But all of these are based on the assumption that the transcription factor, the regulator, will itself be regulated by the same mechanism. So that if I've more of the activator, it's because I've got more of the RNA for the activator. We've already seen that that's not necessarily the case, right? Because there are all these other mechanisms, right? There's very poor correlation between RNA and protein levels. So it's more realistic to assume nothing about whether the regulator itself is regulated level of transcription. And so working with Anima, who many of you know, Tony has actually done some nice work using latent graphical models to actually try to discover some of the regulators that are not themselves present in the transcriptional data. And you can ask Tony for the details of this, but the basic idea is the following. We're going to have a graphical model where each node starts off as being a gene, and the edges represent the correlation, minus log of the correlation between the expression in some large compendium of data. And so Anima developed these kinds of approaches working with a lot of other kinds of data, not gene expression data. Here's an example of hers looking at co-occurrence of words in text.
Actually, that's not unrealistic.
And so you might imagine a lot of correlation in the occurrence of these words in text, which could be better explained if you introduce some hidden nodes in the data, right? So all of these teams on the right-hand side are British football clubs, soccer clubs. All the ones on the left-hand side are American baseball clubs. Those terms, baseball and soccer, might never occur in the data set, but if you put them in, you'd have a better understanding of what the relationship is among all of these nodes. And so we want to do something similar with genes, where these stars would be the regulators, which might themselves never appear as changing in the data set, but would explain why the black things, the genes, are changing. Okay, so her method in an instant. She starts off with a maximum spanning tree, where the nodes, again, are the genes. The edges are weighted by the correlation, minus log of the correlation. And then does a local search to try to figure out what the relationship is among the nodes in a local neighborhood, and whether it makes sense to add these hidden nodes that would represent the parents of these child nodes. And you can use Bayesian information criteria to decide whether you've added too many of these latent variables or not. So in her kinds of problems, here's an image, a problem that she had where they were detecting objects and images, and so each node here represents an object detected in an image. And if you zoom in, you can see they've added a hidden node here that explains why you've got a co-occurrence of desk and book and screen and clock and monitor. And presumably this is an office setting, whereas there'll be other settings where you have sofa and armchair and television that the hidden node might represent a living room setting. And so what they did working with Tony was to try to apply this now to gene expression data. So we've got some vast compendium of gene expression changes in yeast. Again, each row here represents one of the genes in yeast, and each column represents some condition. The yeast were exposed to different types of environmental perturbations. So they look for the most variable genes across this data set, used exactly the same approach to build the latent tree. The tree itself, a little hard to understand what it means, and so some of the hard work here was how to interpret this and figure out whether your latent nodes are actually telling you something biologically meaningful. And so the approach that they took was to look at the neighborhood around each latent node, look at the genes that are in there, and see whether that grouping of genes together corresponded to genes that we know to be targeted by the same transcriptional regulator from the experiments that we had done a long time ago to actually physically determine that. And the results are pretty good. So if you compare the results that they get with the latent tree graphical model to the approaches people have gotten previously by looking just for self-consistency in the data, you look at a bunch of stress conditions which have been well studied, and the latent nodes that the latent tree graphical model comes up with correspond to transcriptional regulators that we know from other data are actually regulators of stress response.
This is you building this tree over the correlation structure of the RNA activity?
That's right. And then the latent nodes, we believe, should represent transcription factors. So we ask, do they line up with known transcription factors? And so they do, and they map up with known transcription factors that actually are regulators of the stress response. Whereas this other kind of approach, which looks for self-consistency, it found some genes that seem to be consistent. Oh, maybe this is a regulator that turns on these genes. That's why it's red when they're red. But the identities of these things do not map at all to what's the known biology of stress response.
And do these transcription factors, I mean, do they overlap a lot, or is it actually reason to believe this is tree-like for the things we know?
No, it's probably not tree-like. Yeah, so that's an approximation that we're making. Right.
How good? So it's like one thing. There's like, because it doesn't seem like you're that computationally constrained here
Right.
when you're working with these tree models. Like if you just try other ways, because there's a whole bunch of kind of consistent ways to build these trees if the model's right, and if it's wrong, like it'll tinker. So like how stable? Because you're getting kind of good things here, but if you run it, will you get other stuff that looks interesting because it's sort of not quite a tree?
Yeah, so how stable is the solution? Maybe I'll turn this one to Tony. Do you have a...
We've toyed with the extremes and not stability in narrow regions, so I think that's an open question right now.
Okay, well,
Unrelated question. What do the red edges between these internal nodes mean? I mean, what are they?
oh yeah, but so between these nodes, these actually represent known physical or other functional links among the proteins. So the reason that we get a lot of these hidden nodes are highly connected we're close to each other in the latent tree structure, and that makes sense because the proteins themselves are known to interact with each other.
And what is like a depth to? So one level up is just the transcription factors, but are there transcription factors that affect other transcription factors? Or?
There are, yes.
Okay, and are you actually discovering things sort of beyond level?
So we get rid of the tree structure when we actually do the analysis. So we look for things in the neighborhood of the latent nodes to try to interpret.
You mean all the way down to the?
Yeah.
So you ignore any kind of hierarchy among.
That's right. And I'm not sure that there is too much biological meaning to the hierarchy you get out of the latent tree for precisely the reason you're talking about, because it's not likely to be a tree structure.
A way to extract some nice correlations.
Exactly.
This is a toy example. The previous one that you described was like this 51 out of 90 latent nodes being sort of matching with the current thing. How many of these red nodes were there in that?
So this is not a toy example. This is a piece of of that bigger one. Yeah.
And how big was the overall instance of here?
We started off with, I think it was 500 genes, 5,000 genes. No, we started off with 1,000 genes. We threw out 5,000. So we started off with 1,000 genes across 500 conditions.
1,000 genes across 500
Conditions. So the matrix is 1,000 by 500.
So what do the conditions mean in the tree that's being ignored? That's ignored.
That's where the correlations come from, right?
Okay, that's the samples. I see. So
Samples, sorry.
A different correlation. I see.
All right, well, we can discuss that more later, maybe. This kind of approach, though, takes 500 samples, takes large, you need a relatively large matrix to be able to do this.
Small sample size.
For biology, it's a very big one.
Right, but for significance issues are important here. Unlike
that's right.
Sort of ML thing, 500 is nothing. Right?
That's right. Yeah, exactly. So if you were doing this on text mining, 500 would be tiny, right? And so one of the big issues we need to deal with is a lot of these statistical methods are not going to work under the kinds of settings where we are trying to answer biological questions. So if you're dealing with a disease model, you're not going to have 500 different variations or 1,000 or 10,000. You may have 2 or 3. And so these approaches just don't work in those settings. So we needed a different approach for that. I'm going to just very briefly try to explain what that is. It's called using epigenomic information. And the idea is that this is some cartoon of the region preceding a gene where these regulatory proteins are bound. We don't know what they are, but there are certain things that change when these regulatory proteins bind. They're not changes directly to the DNA, but they're changes to things that are wrapped around the DNA, which is why they're called epigenome, around the genome. And there are individual experiments. So some of you may have heard some of these terms, maybe not. Histone modifications, DNA methylation. The important thing is that there are very efficient experiments that can measure these epigenetic modifications, which are indicated here in the cartoon by these arrows, in a single experiment across the whole genome without having to know what the identity was of the proteins. And so if we do this and we measure systematically where all these epigenetic modifications are, then we can look specifically in those little pieces of the genome. The genome, human genome, is about 3 billion with a B bases long, but there might be a very, very small fraction of it where we find these epigenetic modifications. And so we can then go and look at the sequence of those regions and try to figure out if those sequences give us some indication of what the proteins were that are indicated here by the dashed line. And so we have various different approaches. A lot of them are based on some sort of correlation approach where we might expect stronger copies of a match to some sequence pattern to be occurring in places where there's a bigger biological readout. I won't go through the details of that. Then we can go through and actually do experiments. So we make some prediction that this yellow protein is bound there. We can actually do an experiment where we try to physically detect whether that yellow protein is bound to those regions using massively parallel sequencing approaches. And we can get pretty good results. So a grad student in my lab, Chris Singh, recently had 2 papers where he showed in a neurodegenerative model of Huntington's disease that he could use expression data and these epigenomic features to identify what the regulators were. And this does not require hundreds of samples. It can be done in this case with 2 samples. I mean, a little more than 2 for biological replicants, but we're talking about order of magnitude. Okay. All right. So we've shown, I've shown you a couple different ways to try to figure out what these regulatory proteins are that are binding to the genome. Now we have all the information we need to try to put together the signaling all together. So again, we're trying to reconstruct this pathway. Some external signal starts of that whole phosphorylation cascade that you learned about in the guide, instant guide to molecular biology. That sends some signal to the nucleus. These proteins change in their binding, their activity, and that makes RNA changes, right? And so Carol Huang, who was a graduate student in my lab at the time, is now a postdoc at the Salk, recently published a paper with us where she showed how to combine all sorts of different kinds of data to understand a brain tumor called glioblastoma. This is the one that killed Senator Ted Kennedy, for example. And it's usually detected at a very late stage. There aren't really very good therapies for it, so most patients don't survive more than about a year after detection. So the hope is, again, and we're not there yet, the hope is that these kinds of very systematic measurements could lead us to come up with new therapeutic strategies that you might never have come up with through the traditional biology approaches. So Carol focused on a particular mutation that occurs a lot in the patients who have the worst prognosis in this disease, and it's a mutation in a protein that spans the external internal boundary of the cell surface membrane, and it changes signaling through the cell and actually changes one of the activity of one of these kinases, the proteins that add these phosphate groups. And so she used data that one of our colleagues had collected on which proteins are changing this phosphorylation. And she had cells that express the mutant form of this molecule or an alternative form that isn't a kinase because it's been changed so it can't function that way. So she had data on which proteins change in that phosphorylation signal. And she also had data on these epigenetic information about where proteins are likely to be bound. And she did a similar kind of regression approach to what I described previously to try and infer which the regulators were. And so the goal was to combine all that data, make this network model. Now, the one thing I haven't explained is how we're going to connect the nodes in this network are going to be proteins and genes. Where are the edges going to come from? And here we use databases of physical interactions that have been detected by lots and lots of different measurement techniques. Some of them were very focused on particular proteins or genes, and some of them were very unbiased by any preconceived notion about what was interesting. And just systematically measured, do I detect protein A physically interacting with protein B? So we have tens of thousands of these edges and this giant thing that sometimes people call the interactome, because biologists love adding the suffix "ome" to anything they can think of. So we have the interactome. Okay, so we've got this interactome, and we've got the experimental hits. These were the list of proteins that changed in phosphorylation, the list of genes that changed in expression, the epigenomic data.
These measurements, and that's probably not a good model, or
they are pairwise predominantly. That's right. You can reconstruct complexes. There are other kinds of experiments that do detect complexes. So there are ways of doing it, but primarily it's in the database as pairwise interactions, and that's how we use it. So what happens if you ask whether the experimental hits can be connected together by the detected protein-protein interactions in the interactome? So good news and bad news. The good news is you can connect things. The bad news is that it looks pretty much like what you started with, right? So you get one of these giant hairballs at the end. And the reason for that is that the data consist of a lot of false positives and false negatives. You've got false positives in these data. You've also got false positives in the interactions and false negatives. So if you try to force everything to be connected, you get a lot of meaningless information. So the approach that we took was first to try to take a probabilistic view of all these interactions, assign a probability to each edge based on the likelihood that it's real, and then try to find an algorithmic approach to the sense of the fact that our hits from the experiments, the proteome, the proteomic information, the gene expression data, has a lot of false positives as well. So this issue of how to assign probabilities of interaction, a lot of people have looked at this. There are many different approaches. Some of them are Bayesian, which are kind of nice. They say, how often does each experimental technique get the right answer on some gold standard true positive and true negative? And then you can calculate, okay, now I have an experiment that detected interaction. I have an interaction that was detected by particular experiments. What's the probability that's true? There are other techniques that are a little bit more ad hoc, but there are a lot of scoring schemes that have that general flavor that can give us an approximate probability of interaction for each edge based on what the underlying data are. Okay. So this is for example now where we've got some set of gene expression changes as the squares. These triangles are the transcription factors, the DNA binding proteins that we inferred from all that epigenomic data, right, that we believe are turning on these genes. And then maybe we have the changes in phosphorylation that are the yellow circles. And this is some piece of that giant hairball, and we'd like to have an algorithm that could help us find the things that are highlighted with the purple lines, the piece of that hairball that's most likely to be functionally linked to each other by these physical interactions. In order to do that, sometimes we have to throw out certain data points and say, well, I can't trust the connections between this measured phosphorylation change and anything else, because these edges maybe are too unreliable. And when I'm trying to figure out whether to include this, okay, it's some long chain of interactions, but maybe these are all really high confidence and I should include them, or maybe they're low confidence and I shouldn't. So we want an algorithm that's sensitive to that approach. And what we ended up doing was using this prize-collecting Steiner tree algorithm. And the core idea is this. We're going to give every node a prize if it was detected as changing in the gene expression or the proteomics. There's no prize associated to genes that were not detected as changing. And we'll give then prizes to the transcription factors as well. And we'll assign costs to each edge based on these probabilities of whether they're likely to be true or not. So you'll have a high cost if it's a very improbable edge, not likely to be true, and a low cost if it's likely to be true. And the algorithm is trying to decide every time it includes a node in the final network, does the prize that it gets to keep by including this node justify all the costs for the edges, right? So if it's a big prize and low cost, then it definitely gets included. And if not, then there has to be a trade-off. And so what you end up optimizing is you minimize the number of prizes that are not included in the final answer and the cost of the edges that are included.
So you're trying to build a tree then?
You necessarily then with this approach will end up with a tree. Now what we actually end up doing with it afterwards, we relax this tree requirement by taking a lot of suboptimal solutions, we add back edges. So this algorithm gives us a tree, but we don't think the biology is a tree. Yeah. But what it then does is you go from having one of these hairball diagrams. So this is what you really get when you take the data from that glioblastoma example and just look at the nearest neighbors. And you get thousands of hits. And instead, this is the network that we get with the prize collecting Steiner tree, where everything that's outlined in red had a prize, and the nodes that don't have any red around them were what are called Steiner nodes. They're added just because the algorithm says they're needed to connect up the things that do have the prizes. You look like you have a question?
Yeah, so the Steiner nodes, where are...
What are the boxes, the big boxes? What are they?
These are our biological interpretation of what we found.
Let me just make sure. So the triangles, those are the gene expression.
These are the proteins that we think explain the gene expression.
Okay, and then the circles are the
Steiner nodes.
But what do they correspond to?
They correspond to proteins that we didn't detect anything about, but they are needed to physically link the ones we did detect to each other.
But you know what? So you don't know what those proteins are. They're just nodes you put in.
In some cases, we know from the literature what they are. In other cases, we don't know, but we know that they have physical links.
And after the fact, you can tell us what happens?
Yeah, so exactly. All right. So when you get a network like this, there are all sorts of statistical tests that we can do to try to figure out if it makes any sense based on prior knowledge. That's good, and it did some interesting things that I actually didn't expect it would work out, some of which we were forced to do by reviewers. But really, if you just are recapturing the prior data, that's not the goal of all this, right? We really want to try and find something new. So we asked some questions about some of these Steiner nodes. For example, I was very suspicious of this Steiner node because it's a protein that's been studied a lot, has a lot of interactions, and it could be there just because of the hub in the network. And so if you've got a hub in the network and you need to connect the things with prizes to each other, you're going to naturally have a certain preference to picking up hubs. So we did an experiment where we physically measured what this protein did. So it doesn't bind directly to the genes, but it binds two proteins that bind to the genes. We detected where it was bound in the genome, and then we asked whether the genes which it's bound are also places where that epigenomic signal was occurring. Right? We had measured these things independently, and it turns out that this protein, which was not directly detected in any of the experiments, but the algorithms told us was relevant, was highly statistically correlated with this signal that was actually in the samples that we were looking at. So that was good. What we really wanted to do, though, was see whether we could actually come up with potential drug candidates with this approach. Remember, this is data from a tumor cell. So we ranked every protein in the interactome by whether it was in our core solution or very close to it, highly connected to it, or whether it was very distantly related. So we have a ranked list of every protein in the interactome. And we chose a few of the high-ranked things that were really tightly part of our network, and the things that very low-ranked that seemed very distant. And then we used chemicals that block the activity of those proteins. And we asked whether they block the growth of these tumor cells. And for things that were very distantly related to our network, we didn't do very well. For things that were core in our network, we did very well. A lot of these hypotheses held up, some of which I didn't think were going to work in a million years. So this is a brain tumor. One of the things that was really highly ranked was the estrogen receptor, which would have no apparent relationship to brain tumors. But when Carol did the experiment, she found that tamoxifen, which is a compound that's used to prevent breast cancer, actually blocked the growth of these tumor cells very effectively. So again, this is all in vitro. We're not curing cancer yet. We're curing cancer cells in a plastic dish. It's important to make that distinction. But it does appear that we can take a lot of data that's unbiased by preconceived notions, build a model, and use that model to guide experiments that are going to be much more accurate ways of finding these novel targets than just chance or our preconceived notions.
So just to understand what these graphics mean, so having the black bar equal to the white bar is sort of bad news, and if the black bar is suppressed a lot, that's good news, right?
So that's mostly correct. So we have two kinds of tumor cells here. One that have the specific mutation and others that are tumor cells but don't have that specific mutation. So if you knock down both the black and the white bars, that's good. But it's even better if you knock down the black more than the white, because then it's very specific to this particular mutation.
Fifth column here would be even better.
1, 2, 3, 4, 5. Yeah, so that's obviously very, very strong blocking of the growth of these. Yeah.
So is there a prediction? Is the model just saying this is an interesting thing to look for, or is there actually a prediction of which one's going to be higher? Like,
Yeah, so that's a really great question. So these current models say this is an interesting protein to study. It doesn't tell us whether we should activate it or inhibit it or something else. One of the things that we're working on now is actually trying to convert these kinds of models into other models that are makes much more quantitative predictions. I have a slide on this later, but the basic idea is that a lot of those kinds of models can't work on a genome proteome-wide scale. You need to have it boiled down first to some couple of dozen proteins to look at. And we think this is the way to boil it down to the couple dozen proteins and then feed it into these other algorithms that are a little bit better developed for making quantitative predictions.
Have you tried doing this on some case where the cure is known, and to see if your things? I mean, how do you know that somewhere in these?
Yeah, so that's what the stuff I skipped over. We compared what we found to a lot of known data about glioblastoma, and so we looked at the ranking induced by our genes compared to a list of genes that people had previously said were relevant to glioblastoma. So glioblastoma is not a completely unknown disease. People have a sense of what's important and what isn't. And so we recapitulate that.
I was more curious if you know that there's not a better remedy, some other thing that you could have tried to, some other gene that you could have tried to attack further down, but you looked at the top 27 and you found some very significant factors.
So do the whole experiment in a system where we really know how to cure the disease? No, I mean, it sounds like a great idea, but in terms of the human and real cost of doing these, it's not really practical. But yeah, that would be interesting, I guess. I guess I only have a few minutes, so I won't try to go through all of this. I'll skip over we've been able to apply different kinds of data and come up with different ways of ranking things in these networks to decide what to target. One thing that Tony's been working on that I think is really exciting is what to do when you've got lots of information about different patients who you think have a common disease, but the actual data for those patients looks very different one from the other. And I'll skip some of the motivation. This is, I think, a very informative slide. So there had been a lot of excitement about trying to sequence lots and lots of tumors in the hope that if you look at a large enough population of tumors, you'd see certain mutations occur over and over again, and those must be the mutations that are really driving the tumors, those other mutations just occurring along the way. So a huge amount of money got poured into sequencing tumors. But what people found really disappointed them, because they found there were only a handful of mutations that occurred frequently. So this is a whole bunch of genes, and the list could go on and on and on. And then the frequency which they occur in tumors. And the ones that occur frequently were ones people already knew about. And then most of the other genes mutations occurred very, very rarely. And so there wasn't really a lot of immediate biological insight that came from these. But each patient tells a story here, right? Each patient had some mutational process that ultimately led to the disease. And so the way we've been thinking about this is you've got some network, and some piece of that network gets, if some piece of that network gets perturbed, then the cell turns into a tumor cell. And so there's some probability that any protein in that network will show up in a final patient. But there's no guarantee that the same one would, right? So a patient will be a combination of mutations that occur with some probability over some network, and then a bunch of other mutations that occur by chance in that network or in other networks. And so the final network for all the patients might look very different, but they might have pieces that are common. And so we can take the same prize-collecting Steiner approach that we had before, where before we were saying, does adding this node make sense in terms of the edge cost and the information that this is changing in that sample? And now we can say, well, what do we know about this from the other samples, from the other patients? So if I know the ones in green were mutated in some other patient, then I would have more confidence in adding this node to my network, even if the edges are not as reliable. Whereas if there were no other mutations, then I might have less confidence in adding that piece. And Tony's been able to show, and I'll have to skip all the details. Well, I'll just go through this bit on the equation. So we had this equation that I showed you before, where we optimize the network to minimize the cost of the edges that are included and the number of prizes that we leave behind. This is for a single tree or forest. And now what Tony's done is said, okay, now I'm going to do my optimization across a whole bunch of different patients. This represents this optimization function. And then I have another term that couples the nodes that are included across all the patients, that tries to bias me to have those green nodes that were shown before. And so we have a whole bunch of patients. We map the data onto the networks. We learn the individual trees or forests. Then we see, okay, certain nodes, how frequently does each node occur across all of these? And then we can bias the network for each individual in a soft way to prefer pieces of the network that have shown some evidence in other patients but not require it. And I'll skip all the details, just to show a few things on cancer patients. So this is from a giant sequencing effort of breast cancer. In yellow are the mutations in an individual patient, and in green are ones that occur in some other patient. You can see how dense this is in green. So even though the individual patient might be very sparse in this network, mutations are seen in this piece of the network in lots of other people. You can see, for example, that this kind of approach can pick up mutations that we know from other data to be relevant, but would not have been picked up if we analyzed the patients individually without this coupling. Okay, so just to sum up, we think that this kind of problem where we integrate many, many different kinds of data by looking for relationships among them, the physical links among the data that are generated by different techniques will be more powerful than simply trying to analyze in terms of known pathways, and that we can actually find novel stuff, new approaches to diseases by this kind of integrated approach that we could not have done previously. And so we're working with a whole bunch of problems in our lab and with collaborators from everything from pancreatic cancer to plant metabolomics to see if we can push forward to really develop new approaches to important biological problems. I tried to mention all the people who were directly involved within the talk. Obviously, a lot of this benefited from great interactions here, and also with Ricardo Zucchina at the Politecnico in Turin. And we have a lot of great collaborators in MIT in the area who are suppliers of great biological insight as well. So I'll stop there. I think I've run over. Thank you. Yes?
I wanted to ask about the shift that you mentioned in the beginning, cartoons, to use your term, to more computational models. And I read this article last summer, I think it was on scientists who model crystal formations. And they were talking about how they used to build physical models, and then they just stopped, sort of, and then now they do it computationally. The article was sort of talking first about the relationship to the crystals that the scientists formed, and they felt like there was sort of a loss there, like in understanding the crystals, but then also a sort of pedagogical loss, and they found themselves really struggling to explain crystal formations to students without making that intermediary step of building a model. And I was struck by that even in your talk, where it was like we sort of had these moments, and like you and Sean were better at like expressing them, of like, wait, what does this mean, and what does this mean, and like how do you read these symbols? And so I guess I was just wondering if you felt there were any similar losses in this transition from cartoons to computational models.
That's a really good question. I don't know if you were picked up by the mic, so I think that's the recording. So I'll just say that the question had to do with whether there's a loss in understanding when you go from the cartoons, as I call them, to these computational models. That may depend a lot on how you were trained. So I got my PhD in a biology department, but I used to hate signaling because I could not make heads or tails out of it. It just seemed to me, too complicated, just this morass. And now I'm doing work on signaling, but it's precisely because I think these computational models make sense of the data in a way that previously the cartoons didn't. People who loved the cartoons probably missed them. I personally won't miss them at all. At some level, though, you do want to be able to understand what the models are predicting, right? And maybe the analogy should be to other kinds of engineering, where when people are designing airplanes, they had to go away from building balsa wood models to, computational models. I'm not an aerospace engineer, but I imagine you still need some training in the balsa wood models to really understand what's going on, right? So how do you train people in this field is something that's important. It could be, though, that the modeling takes now a more important role. So you could have a computational model where then you say, well, what happens if I perturb this? And if the models really are predictive, then maybe you can develop better intuitions than you could in the old-fashioned way. But it is something we struggle with, how to best convey. What does it mean to understand what's going on, right? Is understanding enough that you have a program that gives you the right answer, or should you have an intuition of what's going on? Yeah.
Like, somehow these computational models, they're really bad, right? And somehow, because you see this, like when we play on these latent trees, right? You never get deep, kind of really deep trees. What you end up doing is trying to make these internal nodes where all the observables hang like right off of it. So, which is sort of the point here. You're kind of ignoring any deep structure, the transcriptional stuff. And so I don't know about the other one, but I suspect you wouldn't buy anything like too deep in these trees that aren't kind of lots of observed stuff hanging off. I don't think we would find it.
Actually, I would disagree with that. So in the Steiner nodes, because the links that we're getting are physical links, the structure actually is more informative. So you do get signaling. You remember those cartoons had this chain of events. So those chains of events are actually physical links that are there. So we don't rely on statistics to reconstruct those. We actually.
But the chains, like, sort of observed stuff is hanging off them, like, all the way through, right?
But they can be internal as well. So it doesn't just have. They don't have to be leaf nodes. They can be internal. The observed stuff can be internal nodes.
In observed stuff, I mean, stuff that you can measure is kind of hanging.
Stuff that you can measure can also be in the middle of it, right? Remember the one with the red boxes. It wasn't all leaves.
There's no notion of middle, like somehow.
Oh, we've gotten rid of. I mean, so this is not a tree, obviously, right?
Yeah, that's what I'm saying, and then you fix this because you explicitly don't make it a tree.
Right.
Like somehow it's more like you want some reasonable way of capturing local interactions that aren't a mush, and this seems to come up all the time. And using tree more as like a computational heuristic than.
That's right. Tree is purely a computational heuristic for us.
And sort of running into problems with that like all the time.
Yes.
You kind of see, and so just intuition for how, because like for the latent tree stuff, like working with Nino, like with an email, like one way to these data sets is you try to build a tree. Another one is you try to do something much more shallow that's overlapping. Right. Because of those interactions there, and showing what intuition you have to sort of fix these models in a more reasonable.
Right, so we throw away the tree structure. In fact, the way I think of this is, it gives me a bag of nodes, right? And then understanding the relationship among that bag of nodes is a separate biological problem. And in fact, we've been exploring trying to do that to try to reconstruct what the right edges are using other kinds of data. So if I start off with this bag of nodes, then maybe I can reconstruct what the right set of edges is if I had data, say, on the time dynamics of these nodes. And so that's something that Tony's been working on with collaborators as well. Time dynamics is an important component. There are other kinds of data as well that we might be able to then roll in. So I see this as a way of finding out where to focus our energies and then choosing the right experiments to reconstruct it. So the time dynamics, you could then do very focused perturbations of a whole bunch of these nodes once you have it down to some manageable set. And maybe you could reconstruct a logical network. Just sort of, I'll give you my view of how all these modeling approaches fit together. Okay, so I think about these modeling approaches as whether they're good for systems where you have known components or unknown components, and whether they work on physical relationships or statistical relationships. So if I've got some giant data set that includes all the proteins and genes, then maybe I'll do regression or clustering, and I get some statistical relationship. There are other approaches that people in biology like a lot, well, not biology, but biological engineering like a lot, like differential equation-based approaches or logic models, where if you've got a very small system, you can make very quantitative predictions about what's going on. So we've been doing LIMS over here. It works on physical relationships at the genome and proteome-wide scale. And what we're hoping is we can take all this kind of omic data. That'll lead us to which pieces to work on. Then we could feed it in to reconstruct some of these differential equation-based models or logic models and so on. So yeah, these models don't really explain, they don't make quantitative predictions in a way that we're going to need to actually be able to solve serious biological problems. But they do help us figure out where to focus.
Thank you again.